The birth of an offspring is perhaps one of the most important and joyous events in the life of a sexually reproducing organism. This event, however, is a culmination of events such as the development of the zygote and embryogenesis. Both these events, incidentally, are the mainstay of post-fertilization, the third stage in sexual reproduction. Did you know that every sexually reproducing organism begins his or her life as a zygote? Depending on whether fertilization is internal or external, the zygote is either formed inside the organism or outside in an external medium such as water. After its formation, the development of the zygote is largely influenced by its environment. After its formation, the development of the zygote is largely influenced by its environment. Take the case of organisms such as fungi and algae, whose zygotes are formed either in water, rocks, or soil. These zygotes develop thick walls that are resistant to desiccation as well as environmental damage. However, an unfavorable environment forces the zygote to undergo a period of rest and start storing nutrients before germinating. The environment also influences the germination of the zygote. Apart from the environment, the development of the zygote is also influenced by the life cycle of an organism. If the organism has a haplontic life cycle, as in the case with Chlamydomonas, you will notice that the zygote divides meiotically and forms haploid spores. However, in the case of a fern, a pteridophyte with a haplodiplontic life cycle, the zygote divides mitotically to form a sporophyte. After the initial development, the zygote slowly starts metamorphosing into an embryo through a process known as embryogenesis. During this process, the zygote starts to divide mitotically, which results in an increase in the number of cells. Simultaneously, the zygote also undergoes cell differentiation, a process during which groups of cells go through modification to form different tissues and organs. For example, the cells of an embryo differentiate to form nervous tissue, which later develops into organs such as the brain or spinal cord. Embryogenesis, which occurs in almost all animals, can take place inside or outside the body of the female. In oviparous or egg-laying animals such as reptiles and birds, the process of embryogenesis takes place outside the body of the female, that is, in eggs laid by these animals. The eggs, covered by a hard calcareous shell, are usually laid in a sheltered place such as pens and treetops to keep them safe from the prying eyes of predators. Once the incubation period is over, the young ones hatch from these eggs. In the case of viviparous animals such as mammals, the process of embryogenesis occurs inside the body of the female. The embryo derives all its nourishment from its mother and begins to develop into a young one. After attaining a certain stage of growth, the young one is ready to emerge from the body of the female. Did you know that the young ones of viviparous animals have a better chance of survival than the young ones of oviparous animals, as the body of a female offers better nutrition and protection than an eggshell? Apart from animals, the development of the zygote and embryogenesis occur in flowering plants as well. Interestingly, the formation and subsequent development of the zygote inside the ovule triggers the withering of other parts of the flower, including the sepals, petals, and stamens. The pistil, which contains the ovule, however, remains attached to the plant. During embryogenesis, the zygote develops into the embryo, whereas ovules develop into seeds. The ovary, on the other hand, develops into a fruit. The fruit is covered with pericarp, a thick wall that protects the seeds and the embryo. After the fruit is consumed, the seeds get dispersed by wind, water, or animals. These seeds later germinate into a new plant. Thus, in both plants and animals, the formation of a new life hinges on the development of a zygote and embryogenesis, the two post-fertilization events.